Okay, so good afternoon, everybody. My name is Simon Olsen. I'm the PI of the AI for Science group at Chalmers, and we are organizing this AI for Science seminar where we are inviting excellent young researchers from industry and academia to present their work. This is primarily focused to give a platform to more junior researchers to allow them to establish themselves and have a broader audience, both at Chalmers and internationally. So we are very happy to have Kevin Yang here for the second installment of the seminar series. Kevin Yang is a senior researcher at Microsoft Research in Cambridge, Massachusetts in the US, and he's working on machine learning and applying these modern machine learning methods in the field of biology, in particular protein sequences, as he will be talking a bit about today. He did his PhD at Caltech with Nobel laureate Frances Arnold on protein engineering using machine learning. Before joining Microsoft Research, he was a machine learning scientist at Generative Biomedicines, a startup, where he was working on optimizing protein sequences. Before graduate school, Kevin was a math and physics teacher at a high school in Inglewood, California, through a Teach for America program. And we are very happy to have you here today, Kevin. Please go ahead and share the screen and take it away.
Thank you. All right. Cool. So today I'll talk about multimodal machine learning for protein engineering. The multimodal part is mostly aspirational still. A little bit of background. Proteins are biological actuators. So each of your cells contains between 1 and 3 billion proteins each. They're responsible for structure, metabolism, and signaling. So things like IgG, which is immunoglobulin, it's part of your immune system. Hemoglobin carries oxygen in your bloodstream. Insulin regulates glucose levels in your blood. There's nasty ones like the coronavirus spike protein. There's fun ones like luciferase, which makes red bloods glow. And all that diversity and function arises from chaining together 20 building blocks called amino acids. And they're amino because they all have an NH2 group on the left end in these pictures. And then in the right end, there's that carboxylic acid group, the COOH. And then in between, there's a side chain. And that side chain is what makes any of these two acids different, and it gives them their different properties. So during protein synthesis, during translation, these are newly born proteins are put together one at a time, and then they come out of the ribosome and eukaryotes, and they fold into 3D structures, and that's their functional form for most proteins. So why do we want to design proteins? A few reasons. One might be new chemistry. Here I'm showing a new to nature reaction using an enzyme to make carbon silicon bonds, which is a reaction known to organic chemistry, but previously was unknown to nature and to enzymes. You might want to understand function. Here's that coronavirus again. Maybe you want to use a protein as a therapeutic. This is asparaginase, which breaks the amino acid asparagine down. And it turns out that certain cancers can't make their own asparagine. So if you break down all this asparagine floating around, it kills off the cancer much faster than it kills off anything else. So it's a chemotherapy drug. Or maybe you want molecular tools, such as green fluorescent proteins shown here, which people use to dye all sorts of biological things so that you can see, makes them easy to see and makes it easy to see what's going on. So ultimately, the protein engineering problem comes down to, we know that the sequence here, I'm using a letter to represent each of the 20 amino acids. And we can represent the protein sequence for primary structure as this is a sequence or string of letters. And that determines the function. With AlphaFold 2 nowadays, we are getting pretty good, although there are still a lot of edge cases and we still are not good at dynamics and ensembles and things like that. And then that structure mediates function. But what we actually care about is flipping it. So given the function we want to put out this sequence. It turns out that if you, and this is really hard, and it turns out it's so hard that if you figure out a way to get around it, they give you a Nobel Prize. So this kind of algorithm to sidestep the fact that we don't know how sequence determines function is called directed evolution. And the basic idea is you take some parent with a measurable amount of activity for the thing you want. And I'm showing that in the landscape to the right. It's got some measurable amount, but not very much. So then you make some random changes, just like taking little tiny steps in random directions, and see what happens. You measure the results, and you take the best direction and take a greedy step. And you repeat, and hopefully you get somewhere good. And this is fairly successful. You can design a lot of things this way, as long as you have one apparent that it gives you a good starting point. That's measurable activity, and it really helps if you have a high throughput screen. And a very low bar for high throughput is at least 100 measurements a week. So machine learning enables the same sort of optimization, but with many fewer measurements. And the idea here is we're going to take those sequence function measurements, instead of just using the best one as a new parent, we're going to take all of that information, throw it into a machine learning sequence function model, and then somehow use that information to decide what to see next, instead of randomly mutating a new parent. As a quick case study, I'm going to talk about making designer channel adoptions. And this project was done with a lot of collaborators, the most important two being Claire Bedbrook and Austin Rice, who did almost all the experimental work for this project. So channel adoptions are light-gated ion channels. You can see I've shown the crystal structure here. It has 7 transmembrane helices. That's those blue loopy things. And the membrane I'm depicting with this is slightly darker gray. All seven helices go through the membrane. In the middle, you see a kind of golden molecule called all-trans retinol. And here I'm going to show that molecule again. And it sits in the membrane, and it's usually closed. But when it's hit with the right wavelength of light, it shifts the retinol shift conformation, causing the protein to open up. And in this case, it allows sodium ions into the cell. The natural function of these in algae is phototaxis, so it helps algae to move towards or away from light. But if you take these and express them in neurons, and then shine light on those neurons, you can cause that specific population of neurons that's expressing these channel adoptions to be activated or deactivated. And we call this optogenetics because it's genetically targeted and it uses light. And with this, you can express channel adoptions in certain populations of a mouse brain, for example, and then shine light in the brain. And you can make the mice do things like forget it's scared of a certain corner of the cage, or it gets shocked, or you can make it turn left or right as it runs, etc. So basically mind control. And if you want a good channel adoption, you might have some engineering goals. For example, you need to have good mammalian membrane localization. So here what I'm showing in the picture is I've attached a tag to the channel adoption, or Claire actually has attached a tag to the channel adoption, so that when it's correctly localized in the membrane, that tag sticks out of the membrane. Then you can bind GFP to it on the outside, wash away any unbound GFP and image it. So you only see green when the channel adoption is correctly localized in the membrane. And you need that because if your channel adoption is not expressed by a mammalian cell, or if it's just hanging out in the cell, it's not going to do what you want it to do. You might want increased sensitivity to light. So you can shine a weaker light, a weaker laser on the brain and still get the same result. Or that means you can not have to completely remove the skull in order to get the light into the correct neurons. You may also want different activation wavelengths so that you can have a population channel adoptions of different channel adoptions in the same space that respond to different wavelengths of light. You might also want different on-off kinetics. So that just means how fast do you get current when you turn the light on? How fast does current turn off when you turn the light off? How fast does the current decay while the light is on? And for example, you might want something that switches off immediately when the light turns off, or you might want something that basically turns on forever, even if the light is off, for different types of measurements. And machine learning here enables optimization with many fewer measurements. So we start with 3 parents. And here, just to illustrate, I'm going to use photocurrent, which is at a given wavelength, a given given strength of light, how much photocurrent can we get into the cell in nanoamps? And the parents are all around 1. So initially, we design a library and we mix and match parts between the parents. And so here we're swapping bits of one parent into another parent. And most of these are going to be worse than the original parents, but some of them actually do better. If we go more aggressive and try to get as diverse a set of measurements as possible, we actually made about 5 times more measurements than shown here, but most of them don't even localize to the cells. And of the ones that do, most of them have 0 photocurrent. However, we do get lucky and see one that's really good, about 2.5 nanoamps. So then we take a machine learning model, train on all this data, and we design some sequences, and they're a little bit better, but still not great. And we put all this data together again and try again. And then this time we get many improved sequences. And then those 2 purple ones are negative controls that are very similar to this sequence here from the multi-block swap. But the model says they're not going to have photocurrents, and indeed they don't. So that tells us this isn't just a trivial design problem. And these engineered channel adoptions can control mouse behavior without having to remove the skull. Here I'm going to show you a video. When the light flashes, the mouse turns left. And we do all this with only about 120 experimental measurements, which took several months to do. So that's kind of how you can use machine learning to optimize proteins. Another paradigm you might think of is directly generating proteins. So this is what an example, this is the kind of paradigm that I just talked about. And the biggest drawback here is you have to have a starting point still for a parent. Otherwise, everything you measure has 0 activity. So even more ambitious would be machine-generated de novo proteins that do the function you want. So the kind of framework here is you start with a bunch of natural proteins and their functions. You train some model, and you take that model and you specify the function you want, and it spits out a protein that would do. So as a quick case study here, we're going to talk about signal peptide generation. And this was done with, once again, a lot of collaborators. 2 of the most important being Zachary Wu on the right there and Alicia Lee. So Alicia helped and Zach both helped with computational work, and the 2 of them together did almost all of the experimental work. So what are signal peptides? Signal peptides are secretion signals. And so some proteins are meant to be made in the cell and then secreted through a channel outside the cell. And after secretion, that signal peptide is cleaved off, and then the mature protein, which is that top squiggly, will fold into its 3D structure and do its thing. So a little bit, examining the signal peptide a little bit more, they tend to be about 15 to 30 amino acids long. They're at the N terminus of the protein. So they start with methionine at the N region, which tends to be positively charged. There's a hydrophobic core that often looks like a helix. And there's the C region is usually polar and ends with one of a few common cleavage site motifs. So they're responsible for extracellular secretion in prokaryotes and secretion from the endoplasmic reticulum in eukaryotes. They're dependent on both the species and protein, and the rules I've just showed you are descriptive, but they're insufficient for generation. So we're going to try something here, which is we're going to pretend that this is a machine translation problem. I'm going to take my mature protein, which is the protein without the single peptide, pass it through a machine learning model, and then encoder, and then have a decoder spit out the single peptide that goes through. And so we train on only about 40,000 examples, actually, it's not that many. And then we take other mature proteins that weren't seen during training and generate examples of what their single peptides could be. And then we try to see if those can trick signal P 5.0, which is at the time was the state-of-the-art method for detecting signal peptides in proteins. So the way signal P works is you pass an entire protein sequence and it tells you the think there's a signal peptide there. And we also compare to some baselines. So hidden Markov model is not very successful at tricking signal P. The heuristic, which is basically design things using the rules I just told you, is actually better than the hidden Markov model. The variational autoencoder, or VAE, is even better. In our transformer, the probabilities are slightly lower, but if we actually examine the VAE sequences, they're very repetitive. There's lots of repeated L's, and that's just because in that overall, M is the most common initial amino acid, and I think K and R are the second two are the most common in the second spot. And in the middle part, the helix, the hydrophobic part, the most common amino acid is L, and you see lots of repeated L's, where the VAE just kind of guesses that over and over again. So they don't look terribly realistic. It turns out that we can also sick these generated single peptides into bacillocellulose across a few different enzyme families. And 79% of the natural single peptides are functional, and 48% of the ones we generate are functional. And the enzymatic activity for generated versus natural ones is comparable. So what this tells us is even with not really that much training data, we can generate functional single peptides. It's kind of a demonstration of this paradigm of to specify the function you want and generate de novo. We're only generating the beginning of the protein here, not the whole thing, but there's definitely work out there where generate the whole protein. So, second here. In both these cases, we haven't had that much data to work with. We've had 40,000 sequences for single peptides, which is a lot in the protein engineering scale. And for the channel adoptions, we only made probably 200 measurements total. And there's other experiments in this kind of vein where just because sequence function data is not necessarily cheap to collect, people are working with only tens to hundreds of thousands of sequences, of measured sequences. Turns out though that in UniProt, there's 250 million sequences, and there's actually bigger databases now. But if we deduplicate UniProt to UniRef 100, you have about 236 million sequences. Cluster those even more to 90% identity, you have 116 million sequences. If you cluster more, UniRef 50, you have 42 million sequences. If you only want experimentally validated sequences, you have SwissProt, which has about 250,000 sequences. If you want structures, you can go look at the PDB. There's about 169,000 of those, versus for most experiments, you're looking at a few hundred. And one way to use the information contained in these sequences is a paradigm called pre-training and fine-tuning. So the idea here is during pre-training, you take your database of sequences and you train your model. And you take that model and you have specific library measurements and you fine-tune the model further. Then you can predict on sequences you care about and get predictive function. Hopefully this is better than just doing the last two steps. A lot of methods people have worked on in this space kind of pretend that proteins are language. And you can do that because if we write down the amino acid or structure or sequence, they look like letters, and we can just pretend they're natural language tokens and use tools originally developed for language, such as Word2Vec, which assumes that similar words or similar k-mers occur in similar contexts. And then you learn an embedding, or an embedding here just means a vector or a bunch of numbers that describes each k-mer. You can use an autoregressive language model with a neural net. Here you start token, and you always want to predict the next token given what's come before. All the way to the end. And once again, you can use that last layer here, that last output before the final predictions as an embedding if you would like. Or you can use a masked language model where you start with a sequence, you corrupt it with special mask tokens or by randomly changing some of the tokens. Oops, wrong way. All right, so you can corrupt it and then pass it through a bidirectional neural network that looks at every position, looks at every other position, and then you try to reconstruct the original sequence, and you evaluate based on the locations you corrupted. And once again, that final layer, final output before the predictions can be a representation of the sequence. So historically, people have used what are called transformer models, or which use attention, an attention module, to do both the autoregressive language model task nowadays and the masked language model task. And that goes for both natural language and for protein sequences. However, I was curious one day whether you really need transformers for this and attention. And the big drawback of transformers is that the runtime scales quadratically with this input sequence length, which and in proteins, about 2% of UR50, for example, 2.5% is longer than 1,022 amino acids. And that includes things like coronavirus spike protein, includes things like CRISPR-Cas9 that are very interesting. And it turns out that you can use convolutions which scale linearly with sequence length instead of transformers for pre-training. And this is a pretty simple convolutional architecture. You have inputs, you embed that input so it's in the D dimensions at each token, run it through a bunch of byte net blocks, which I'll describe in a second, and then spit out your output probabilities. And each byte net block basically has a bunch of layer norms and position-wise feed-forwards and nonlinearities. And the important one is that 1/5 dilated convolution. So you need dilation to increase the perceptive field so that even for very long sequences, every location can see every other location. And we're going to call this convolutional autoencoding representations of proteins, or CARP. And we see that in the pre-training task. So we can show CARP versus ESM. ESM is a popular transformer protein mask language model. We show that across several orders of magnitudes of model size, the losses are fairly comparable. And ESM1B is kind of their flagship model, and it's got about 650 million parameters. It's training UR50. Our model is also training UR50. It has about 640 million parameters. The difference in performance comes from a combination of their model being a little bit bigger, and we actually don't use the same trained test splits or the same version unit of 50. So those aren't directly comparable, but they're pretty close. And CNNs do in practice scale better with sequence length. So you can see that as sequence length goes up, the ESM goes up much faster. ESM won't take inputs longer than 1022 amino acids, which is why that yellow curve cuts off sooner. And you can run sequences that are very long through CARP and get reasonable losses. All right, but that's a pre-training task. No one really cares if your model can reconstruct arbitrarily corrupted sequence. What we really care about is, will your model help me design new proteins? And to measure that, I actually took a whole detour and did a whole new project, just coming up with better benchmarks to compare models. And we know in other parts of machine learning, there's benchmarks like ImageNet or Superglue, or in protein world, CASP is the big one for protein structure prediction. And all of these have driven remarkable empirical progress in machine learning performance. So for this benchmark, we wanted to focus on mapping from sequence to fitness measurements. So there is some previous work here. TAPE is a previous panel of protein machine learning benchmarks. They have two functional engineering data sets. They call them fluorescence and stability. The problem is that they don't really discriminate well between different pre-training or even non-pre-training methods. So ESM, which I showed earlier, is a big transformer. These are Spearman rows. A smaller transformer trained on much less data does about the same. CPC-Prot is a contrastive learning pre-training method, also does about the same. In fact, linear regression for fluorescence does about the same, as does a small CNN. So a bunch of people and I got together and talked about this and looked around for different data sets, which are called landscapes. And from these landscapes, we're going to choose actually not just random splits, but biologically relevant and challenging splits that test generalization. And then we also train a bunch of benchmarks that people can compare to. So the three landscapes are GB1, and another group back in 2016 looked at 4 specific amino acid positions in the GB1 protein that interact with each other. And they made almost all the possible combinations of mutations to those 4 proteins. So almost all 160,000 sequences and measure them. Then there's AAV, which is a viral targeting protein. And this is mostly out of the Church lab and Therapeutics. So they've made a bunch of mutations targeting the binding interface, up to 39 mutations, including insertions or deletions. And then we also look at a thermostability data set where they've measured thermostability from sequences across tree of life. For each of these, we consider different ways to split the data between train and test. So the most challenging way to split this data is you get to train all the single mutants and you test on the rest. So then to do well, your pre-training or your inductive bias must somehow tell you something about physics or biology to be able to do that. Less challenging is you get the single and double mutants, test on the rest, where you can do the single, double, triple, and test on the rest. The easiest split is you get random sequences to train on and test. And then we have what we call low versus high, where you train on the bottom 80% and test on the top 20%. And we care about that one because a lot of times in protein engineering, the goal is to find things that are better. So you really want your model to be able to predict those. AAV is similar. We do random design, meaning the data we have comes from different data collection steps. Some steps they randomly mutate the protein, other times they've actually trained machine learning models on those random mutations and design new sequences. So we can split either way there, random to design or design to random. We can just do a random split, which is the easiest one. And then same as for GB1, we can go one versus rest, one or two mutations versus rest. Then we did 7 or fewer mutations versus 8 or more mutations. And we can also do that low versus high split. For thermal stability, we cluster, and then we train on everything, but we evaluate on the cluster representatives, and we split by cluster so that you're never testing on things that are super similar to what you've seen before. And then we evaluated a bunch of models, the simple ones being just Levenshtein distance or Blossom 62. So basically just compare things to the wild type and assume that things are further away or more different. You can use supervised models like ridge regression or tiny CNN, or use ESM 1B without any pre-training, which is a big 750 million parameter transformer. Or you can look at pre-training on UDRF 50 or UDRF 90. And the pre-training with transformer only helps on highly diverse landscapes like thermal stability, or when data is very limited, like when you have to train on the mutants and predict the rest. And if you are also building machine learning models for proteins and you want to test on some challenging benchmarks, you can go pull the data for these as nice CSVs, which is very nice when I went back to this project and wanted to compare a new CARP model to ESM. So here I'm showing just one landscape. They all kind of look the same. This is the GP1 landscape. And then for each split or task, I'm showing the ridge regression on the right as a baseline. The purple second from the right is ESM1B, which was pre-computed for the benchmark paper. And then I tried 4 things with CARP. One is pre-train fine-tune. One is no pre-training, but fine-tune the model all the way through. Then green is pre-train, freeze the pre-trained weights and only train the model on top. And then same thing without any pre-training. And we can see that for low versus high, ESM does very well. For the mutation extrapolation ones, 3 versus rest, 1 versus rest, and 2 versus rest, pre-training helps a lot, and the CARP does a bit better than everything else. And fine-tuning, especially when you don't have much data, seems to help a lot. When you just sample randomly, everything does really well, but here CARP also seems to do better than the other ones. So this tells us that on these types of tasks that we care about a lot, convolutions are competitive to and sometimes better than attention. An interesting thing here, though, is this gives us a nice picture. Everything looks nice, but sometimes actually pre-training more or pre-training longer hurts. So on the left here, I'm going to show secondary structure prediction, which is not really a task we care about, but kind of measures how much structural information your sequences, your pre-training has managed to teach your model. And so without any pre-training, we get accuracies depending on your model size between 65 and 72%. And then the better the pre-training loss, the lower the pre-training loss for all 3 models, a tiny 600K parameter model, a medium-sized 66 million parameter model, and the big 640 million parameter model, the better your pre-training loss, the better your secondary structure accuracy. And this is kind of a very pretty behavior that we like. Now, earlier, on the previous slide I showed you for GP1, pre-training seems to help. But the picture is actually a little bit more complicated. So once again, we have a tiny model, a medium model, and a big model. The horizontal lines are without pre-training. And we see that pre-training does, I don't even know what it does. I don't know how to describe this picture. So this is not as well behaved as we'd like. And I don't have access to checkpoints for ESM, but there are other people who've built these similar pre-trained models and shown very similar behavior on different tasks where maybe pre-training a little bit helps, but then you pre-train more and the downstream performance just goes up and down, up and down. So remember I said that we've basically been pretending that protein sequences are a language. I also said the title of the talk was Multimodal Machine Learning for Protein Engineering, but we haven't talked about anything multimodal yet. So it turns out that protein sequences are not language. So quick examples of differences between proteins and natural language. Proteins fold a 3D structure. There's not natural divisions between words and subwords in proteins the way there is in natural language. And if you go scrape the internet for language data, to a first approximation, you can pretend that data is IID. For biological sequences, that's not the case because everything has common ancestors, and so everything's generated by evolution. And then the samples we see are biased by the fact that some things are easier to collect than others. So there's a lot of weird stuff at the bottom of oceans that we don't know about. And maybe if there were life with proteins on another planet, it would look completely different. So it kind of makes sense that we want better pre-training and better machine models. We should take this into account and try to take advantage of the fact of the differences between protein sequences and natural language. And we do this using the framework I call multimodal pre-trained or generative models. So I've talked a lot now about models such as scale sequence, feed them into our model. We can also take structure and feed them in along with sequence. You could also think about substrates or ligands or desired reactions you want to catalyze. Or you can feed in free text. And I'm going to talk a little bit about one example here where we do structure. Did this with Hu Ye, who was a research assistant at MSR. And we're going to combine sequence pre-training with structural information. So if we know the structure and we want to do that same masked language modeling task, meaning we corrupt the sequence with mask tokens and by changing something randomly, if you know the structure, you can actually do a lot better. And we can express this as predicting the masked tokens given the structure and the unmasked tokens. So here I've masked one token of my very rudimentary protein sequence with 2D structure. And now you can get sequence and structure information from its neighbors. And we implement this with a graph neural network. So we take in structure and the corrupted sequence. You look at interresidue distances and angles and turn those into edge embeddings and node embeddings, run them through a message passing graph neural network, and you try to reconstruct the sequence. We have about 19,000 training examples here. And we get a perplexity of 4.96. And perplexity, you can kind of think of on average at every location, how many guesses, how many things am I guessing? And if I'm guessing randomly, perplexity is 20. If I am exactly right every time and put all the probability in the correct thing, then perplexity is 1. So lower is better. If I take CART from before and I train on UNIREF 50, 42 million sequences, and test in the same test set, I get a perplexity of 7.06. So knowing structure helps a lot. You can use several orders of magnitude, fewer sequences, and get much better performance on the pre-training task. But if I take CART pre-trained on UniRef 50, and I just stick its outputs into the graph neural network with its weights frozen, I can get a perplexity of 4.08. So I've dropped perplexity by almost 1 by just freezing those models and sticking it into the GNN. Again, it's kind of ongoing work. So the next, obviously, we don't really care about the pre-training metrics. We care about the downstream metrics. So I'm running all those evaluations. And some future work here for applications of multimodal protein modeling. Obviously, one is property prediction. Even more ambitious, maybe we can shorten proteins. For example, Cas9 is a very large protein. Other proteins we care about for therapeutics are very large. And if we can shorten them while maintaining function, that makes it easier for downstream engineering and drug delivery. And one that's very near and dear to my heart is enzyme generation. So can we learn to generate enzymes that catalyze desirable new to nature reactions? All right. And when I acknowledge that I acknowledged a lot of people as I went, but I did a lot of this work in the BioML group and then I started in England with wonderful colleagues. We don't have a group picture because we mostly started during COVID. And now I see a lot of chat things. So I'll take questions.
Thank you very much, Kevin, for this inspiring and interesting talk. I'll do the virtual clap. I'm sure other people are joining me in the chat. So yeah, there's been a number of questions about whether the benchmark has been published or not, and there's already been an answer in the chat with a link to the preprint. But maybe you can show the slide again with the link to the benchmark. Yeah. I think that would be helpful so people they can go and try it out. I think that would be great. Thanks.
Yes. So yeah, we published it in the benchmarks and datasets track at NeurIPS last year. But the easiest place to find is probably bioRxiv or GitHub.
Right.
NeurIPS doesn't make their stuff easy to find.
Okay, I hope that answers some of the questions that have been in the chat. I have a question. So you actually have two questions. So the first thing is that you talk a bit towards this point in the end, right? Where you basically spend some time talking about how convolutions they perform approximately as well as full attention across the sequence, and you stress this point that dilating the convolutions is important to have a sufficient receptive field. And then you bring in the discussion of structured, and that's important because this can sort of make important physical interactions across that are very far away in sequence, but very close in physical space where the protein actually functions. So how do you square that these sequences or these sequence models with relatively limited perceptive fields of CNNs, that they actually improve the complexity in the last task that you show?
Oh, so the CNN in the last task has a perceptive field of like 3,000 or 4,000. I mean, probably even more. I calculated it once because it's the same dilated convolution architecture. And the way that works is the big model has 56 layers. And the dilation goes 1, 2, 4, it doubles to 128, and then it goes back to 1. So the perceptive field is actually really big.
Okay, so you don't see this as being a limitation. Right. Okay. Great. Okay. So and then I have another question before we let the chat loose. So so you state that sequences are not or protein sequences are not languages, and you give a few examples of this. So where does this leave us with the losses that are currently being used to train these language models? Right, they are sort of motivated out from like psychological tests that are motivated by theory from language theory, right? So where does this put us with the with how we train these types of architectures for protein sequences? Can we take some inspiration from biophysics, physics, or maybe biological sequence evolution to define some losses that are maybe more in line with what the tasks that we want to define? We have some thoughts on this?
Yeah, so there's some interesting work I've seen now. The simplest thing I've seen from a group in Beijing is instead of doing this mass negative following thing on one token at a time, you still mask the same way, but instead of calculating the loss at each location independently, you calculate it for every pair of positions. And the intuition behind that is there's a long history in protein structure modeling and protein evolutionary modeling of saying that we care, yes, about the single amino acid residue probabilities, but also the pairwise probabilities, because all of your epistasis and all your contacts come from pairwise interactions, right? And as soon as you have pairwise interactions, the bigger interactions arise naturally. So it's sufficient to have single and pairwise interactions. And they have a preprint out, but they said they're still working on making it better, whatever that means. Other cool work is like MSA transformer out of Facebook, or instead of acting on protein sequences directly, they act on the MSAs. There's some interesting contrastive training stuff, mostly focused on intrinsically disordered regions, where they're taking advantage of it's really hard to find homologs for IDRs because they change so quickly. So what they've done is they're looking at the regions around the IDRs to get homologs. And then assume that if the regions around the IDRs are homologs and the IDRs also are, and using that for pre-training, which is a very clever idea. And then, so obviously I'm working on combining sequence and structure and some other things. That's not the only work out there trying to combine sequence and structure. There's another work from someone in the Baker lab where they use not a GNN, but a fancier, like a fancier model directly on the point cloud of atoms, but they also, I think they use ESM 1B directly as a sequence model. So it's kind of a emergent thing. And I think as more people who actually know biology get into the field, we'll see more creative things, especially around molecular dynamics or biophysics.
Thanks for your answer. So there's a question from Martin Inquist.
Hi, Martin.
I'll ask him to unmute.
Okay.
Otherwise,
Can you hear me?
You're a little echoey.
Yeah.
Oh, wow.
Okay, I think fake it.
okay, so maybe
I'll
take
read
it.
the question.
Yeah, I can read the
Or you
question.
can read it.
Oh, yeah.
Okay.
What's your view on using AlphaFold predicted structures for multimodal learning as a way of removing the bottleneck of only 19K PDB structures?
Yeah, I think that's an obvious next step to see how much better, and also to see my intuition is it will help, but there's probably some that are bad enough that they hurt, and so there's just lots to explore there with how to use AlphaFold for things like that. And when it comes to downstream tasks too, a lot of the times the downstream thing you care about doesn't have experimental structure. So if you can use AlphaFold there, that's even more helpful. Yeah, I'm eagerly waiting for them to release 3UniRef 100 structures. I keep asking my friends there, and they won't tell me anything because AlphaFold and DeepMind are super secretive.
Okay, so anything else to that question? Otherwise, we'll try with Miguel as well, if he can unmute.
He asked you to read it.
Okay. So I can read it aloud. What is the main difference that must be taken in consideration in training models with biological DNA, RNA, protein structures data versus training models with common data, sales data? So like, what is the main, I guess the point here is the inductive biases one has to take into account?
Yeah, it's hard to say what the main difference is. In large part, one, sales data and images and text are all really different from each other, right? And we don't pretend images are the same as text. Well, maybe nowadays with Transformers, some people do. Even then you adapt them for the domain, right? And we should think carefully about DNA, RNA, protein structures the same way. And yeah, there's some baseline models that you can always try. Nowadays, a tiny CNN is a good baseline model. Linear regression is always a good baseline model. And it seems like a simple transformer model is always a good baseline if you have to compute. But until we have infinite training data, using knowledge we know about the domain should always help.
Okay. So there's a question from Abish. I will go ahead and read this aloud. Would you mind re-explaining why predicting the mask sequence is useful for the distribution P of S masked given the structure and the unmasked?
Yeah, so there is one thing you can use it for, which is, let's say you want to design a sequence that follows a certain structure, you specify the structure. You can kind of do Gibbs sampling and mask and unmask things and slowly converge to things. Or you can, if you know the sequence and structure for most things, you know the structure for just a part of it, and you want to design that part, you can mask that part and try to fill it in. Generally, what I care about less is designing structure, not even designing binders, but designing enzymes and sensors and things like that. And here, we care much less about the pre-training task and more about whether that pre-training task helps us do better in the fine-tuning task. And ideally, you want to see this kind of relationship. I blocked it out. This kind of relationship where the better you do in the pre-trained task, the better you do in the downstream task, which I'm still investigating for the GNN. But a lot of the motivation there was that you don't get that relationship with mass language models of this sequence.
Thanks. So there's a question from Orlit. How useful would using atomistic physical data like QMMD simulation to train proteins for new to nature function generation, so like novel function versus using empirical data, experimental data?
So the answer here is it's very unexplored still. Probably it depends how your simulation is, and there's a lot of work in machine learning, like how do you take advantage of even flawed and biased simulation data or low fidelity data to decrease the amount of high fidelity data you have to collect, right? Or for other properties, there's a lab at UC Davis that's done a fair amount of work on like how can you use simulations to help your machine learning models predict stability. So there's some work there, but yeah, there's a lot to be explored there still.
And there's another question coming in here. Do you have any intuition as to how to generate sequences for enzymes that act on non-native substrates?
Right.
Big question.
That's kind of the big question, right? So to be fully transparent about how far I am on that project, I have a data set that is, we spend a lot of time cleaning and getting as clean as we can the smile strings for the substrates and reactants and any cofactors in the protein sequences. And I'm trying some things with it, but I haven't gotten that far yet. And theoretically, it should be possible. The question is whether the data we have is sufficient for the models we have, right? Yeah, as you can see, I like to share work that's like very recent.
Okay, I think I have another final question, and then I think we can close the session. So you say you sort of made this point that everything emerges from pairwise interactions when you were answering my previous question. So I have a follow-up. So why do deep models work better than parts models? Like empirically, they seem to be performing better on certain tasks like structure prediction with AlphaFold. So why do these models work better? Since like they implicitly represent multi-body interactions and not only pairwise interactions. So do you have any intuition why that is?
So one, AlphaFold is supervised and parts model is unsupervised, right? So it can do whatever random tricks it needs to do to get better performance. Two is that just because pairwise interactions are sufficient to describe everything doesn't mean you can get the correct pairwise interactions from the data you have using this. Statistically, that might be an intractable problem, which it seems to be. And then there's the whole thing about if you have an alignment, you can get pairwise interactions out. How do you take out the spurious ones that are just arise from evolution versus the ones that are actually constrained? And the constraints aren't always exactly one-to-one for various reasons. There's not a one-to-one correspondence between structure and the constraints. So yeah, AlphaFold 2, the short answer for why any of the neural nets might win over a POS model is that they have supervision from the structures themselves. There's also a question earlier from NJ. And is this multimodal protein engineering task published in preprint yet? And the answer is no. CARP should be out. The CNN itself should be out in preprint and in a downloadable form for use in a month or so. And the GNN sometime after that.
Okay, I don't see any further questions. Thank you for your time, Kevin, and for available for questions afterwards. It was very inspiring. It was very nice to have you, and I hope to continue this discussion further via Twitter or email at some point. So take care.
Yeah, thanks for having me. Thank you.